Liberty University Molecular Science 500 Cancer Biology. This is Albert Bawo Jr. Today we're going to talk about cancer biology. There's 4 basic topics. The first topic is cell signaling. Second is cell cycle. Third is invasion metastasis. Fourth is angiogenesis. While we speak of each of those topics, there will be a brief presentation that is verbal over each topic. And at the end, there'll be a brief conclusion. So let's begin with cell signaling. Cancer cell signaling pretty much involves the cell. And with the cell, there's different layers and membranes, and we will discuss how there's a receptor that is on the membrane that receives a protein that connects to the tyrosine kinase. Once that mechanism is activated through the environment, it creates a communication with the nucleus and the DNA. So a tumor cell happens from the mechanism of gene amplification. Gene amplification happens from the replication of the chromosome and the intracellular structures. The second topic I would like to elaborate on with cancer biology is the cell cycle and cell proliferation. In the normal cell, it begins with G, then it goes to the G1 phase, S, G2, M phase. With those phases, you have a continuous process. There's a replication of the contents of the cell. During that replication, it starts with the mitochondria and next the chromosome, and finally there's duplication of the cell. There are checkpoints. The checkpoints occur between G1 and S, also between G2 and M. Now, with genetic mutation, it's important to realize that there's a CDK4/6, and that is a protein that bonds with the cyclin D. From the bonding of the CDK and the cyclin D, the ETF and the ribosome are paired and split off. What is key to realize in this cancer biology and the cell cycle are that there's an abnormal growth. There's an abnormal growth in the normal cell because there's mutations that may have occurred. Mutations that could be a point mutation or a DNA amplification. So with the point mutation, it has happened where there's a message that is misconstrued, and also the same thing occurs with DNA amplification. Once there is an amplification and gene multiplication, there occurs reasons to, or mechanisms to continue to have the cell growth. And those things that contribute to that are what we call growth factors. Growth factors and tumor suppressing or oncogenes. Oncogenes and growth factors contribute to the multiplication of the gene. So with the growth factor, it contributes on one phase and through the tumor suppressor genes. So without control of the tumor suppressor genes and without control of the multiplication of the oncogenes, you cannot stop the mutation from occurring and the proliferation from occurring and the amplification from occurring. So there are dominant genes, the RAS gene that goes inside and affects directly on the chromosomes, and the MYC gene that affects the cell growth, cell survival, and cell activity. There are a number of things that contribute to that environment of the cell that can lead to communication from the tyrosine kinase through the environment to the nucleus. And what I would like to cite is one of the research persons that was included in the initiation and the prolongation of the cell. There's tissue invasion and metastasis. Specifically, before you get to the tissue invasion and metastasis, the requirements of the stroma, the environment, is vascularization and is populated by immune cells and fibroblasts. And these activate stromal tissues to create a reactive stroma. The tumor stroma produces large amounts of interleukin. Now, the third topic that we were going to speak about is tissue invasion and metastasis. With tissue invasion and metastasis, the tumor cell has penetrated across the basement membrane. When the tumor cell penetrates across the basement membrane, it can latch on and go inside of the vasculature, and this problem happens and the tumor cell travels with the blood and the lymph to go to different parts of the bodies, and this is where the metastasis occurs. Tumor cells that go through the basement membrane and is transported through the lymph. The fourth topic that I would like to speak about is cell proliferation and death. With cell proliferation and death, it needs to be regulated. As we were discussing earlier, with the normal cell cycle, there's a driving force, and the driving force is what continues to cause each level of the cell cycle to occur. When the driving force drives it past the checkpoint, CDK, and you can get more replication, and there's no stopping the cell from replicating. And as it continues to replicate, it has the option of going back into repeating the cycle. So the main oncogenes that contribute to that replication are the RAS gene and the MYC gene. The tumor suppressor genes that are not contributing are the P53, the APC, and the BRCA1. With that happening, the replication, the tumor cells has enlarged, and it continues to get large to the point where it needs vasculature. Without vasculature, it cannot survive. So there must be a way for angiogenesis to occur. The mechanism for angiogenesis that requires additional vasculature, excretes protein, and stimulate blood vessel growth. So it degrades the extracellular matrix, and to get the vasculature, there has to be a connection from the vasculature of the patient to the tumor cells. Inside the cell, it is not oxygenated, so it must receive oxygenation. So the problem is that the tumor eventually receives blood supply, which allows the tumor to advance. The goal is to find drug therapy that can be useful to stop it from advancing. In summary, there are 4 topics that we've discussed: uncontrolled cell proliferation, loss of apoptosis, tissue invasion, metastasis, and angiogenesis. The cell proliferation from the loss of apoptosis, and there is no cell death. So ultimately, we need to have a balance that returns back to homeostasis, where there is cell death of cells that need to die, and the apoptosis of killing of cells that need to be to no longer be a part of the body. Thank you.